So, good evening. I will share my screen to you all and we will start in a minute. Okay, so here, so far, what we have covered includes understanding various uh, aspects of environment, how human intervention, how natural processes are responsible for the pollution that might deteriorate its uh, its quality, and that makes the environment which might not be suitable for living organisms and human beings to live. So here we also have studied so far what measures we can take to limit the pollution so that minimum amount of pollution reaches the environment. While if pollution has already occurred, then in that case, what strategies we can have uh, to reduce the pollution or to minimize the waste streams, may it be gaseous stream, liquid stream, into the environment. If it is polluted, another way is to treat that stream. So that can be done by various treatments steps. So far we have looked into air pollution control treatment techniques, and also we started with water treatment strategies, in which we had looked into various household filters, also the domestic or sewage wastewater treatment, and a generalized schematic of how or what sort of treatment steps are involved in any domestic sewage treatment plant or any industrial treatment plant. So now, once we had done that, we started off with discussing each of the process in detail so that you can get to know what the process does and what pollutant is removed at each of the step. So we had looked into various processes like sedimentation, grit chamber, bar racks. Then we had seen equalization tanks, dissolved air flotation, and so on, which were more or less they involved. It was more of preliminary and primary treatment that is given to any industrial wastewater or any uh, sewage wastewater. So I guess it was clear to you all so far. If yes, then we can move forward with the coming class. Okay. So after that, after the like, what is the flow chart or uh, what is the the steps that are involved in treatment. So first it is preliminary treatment, then comes primary treatment, then comes secondary treatment, and last comes tertiary treatment. So these two in the form of bar racks, uh, sedimentation, then equalization tank, coagulation, flocculation. This all we have seen so far for preliminary primary dissolved air flotation, obviously. In the previous classes, now we are moving to the secondary treatment of wastewater. So once all the suspended solids and bigger coarse materials from wastewater, which is the task in performed in preliminary treatment and primary treatment, and after separating those solids, uh, next step is to remove organics from wastewater, which contributes towards biological oxygen demand and chemical oxygen demand. So secondary treatment mainly involves the removal of organics and majorly the BOD part. Okay, so here we will be using some microbial method for the removal of organics, where different methods can be used and microbes will be acting upon the organics to uh, uh, convert the organics, right? To degrade or digest the organics which are present in wastewater. So let's look into various methods. So here we will be focusing on the secondary treatment. Now let's start with what secondary treatment is and what sort of equipments we can use for secondary treatment process for the removal of organics. Not removal, I should say the degradation of organics. So one of the major and conventional process, so that is used in industries involve activated sludge process. So this is one of the most common process, ASP. Okay, so activated sludge process is the most common and like it was one of the most widely used method in industries for the removal of organics. Now what it does here, so here this is the influent where it has, let's say, S naught is the amount of BOD which is present in the influent, and Q is the amount of flow rate. Okay, so S naught enters at a flow rate Q and it goes to a tank which is aeration tank. So here activated sludge process is it uh, uh, it occurs like in the presence of oxygen. So it is an aerobic process. Okay, so degradation of organics majorly can happen in two ways. One is degradation can occur in aerobic conditions or it can occur in anaerobic conditions, right? So here we are utilizing aerobic conditions, which means that oxygen is mandatory for those microorganisms to act upon the organics and convert it into some degraded or simpler forms, right? Or to reduce the toxicity. So here, this is the tank, which is, let's say, having volume V. Now here X represents the concentration of the microorganisms. Okay, it is the microbial concentration inside the tank. Then this is a mixed, uh, you can say this tank, it refers or it is similar to a mixed flow reactor tank. Okay, so what does a mixed flow reactor tank mean? So it is an mixed flow reactor. So it means that there is a continuous inlet of wastewater, continuous outlet of wastewater, and whatever inside the tank it is present or reactor it is present, it is completely mixed. So okay, there will be multiple propellers, pedal mixers, or any sort of uh, um, this blades which can mix or which can provide rotation to wastewater and hence provide mixing. This kind of propellers can also be found. Further, you can add oxygen to this tank in the form of various diffusers can be used. So, these diffusers do? So these diffusers, so oxygen comes out from those diffusers in the form of bubble, right? So that also causes mixing, okay? Because the bubble might burst, and that bursting is it releases high energy, which causes uh, nearby mixing. And since diffusers are placed all around the tank, you can say it is well mixed tank. So since mixing is complete inside the tank by any means through oxygen diffusion, through dispersed diffusers, and through some propellers, whatever the concentration of BOD is remaining inside, the same will be coming out with the effluent. Okay, so here, if let's say if SE is the BOD concentration inside the tank, then outside the effluent will be also having SE concentration. Further, X will also be same. Now let's say that treated water is taken out, is sent to secondary settler. Now secondary settler means because here you have microbes, and further there will be some other suspended solids also present along with the microbes. So total there will be some solids which are present, which needs to be settled or which needs to be removed or separated from the treated water. So in that case, you need a settler. Now this is again a sedimentation process. Okay, so this sedimentation process again, since it is a secondary treatment process, so it is known as a secondary sedimentation process or secondary settler. So settler is the name of equipment. While in case if you are using the sedimentation after the coagulation flocculation, so that will be the primary treatment provided to the wastewater. So that will be primary settler. Okay, so here we have learned two terms, primary settler and secondary settler. So this refers to the sedimentation process provided in primary treatment and secondary treatment respectively, right? So I guess it is clear. Uh, can somebody confirm? Okay. Uh, yeah. So let's move forward. So once you have what happens here, the sludge will be settled here in the bottom part of the sedimentation tank, and water will be in the upper side. So that water is taken out. Still, there will be some microbial concentration. Let's say that is Xe, and here since Se like BOD contributed to wastewater, it is not related to sludge. So Se will remain same, while Xe will change because sludge has settled, right? So here the microbes, they are distributed in two parts after sedimentation. One is recycle, so one is taken from the bottom, which is settled. Another is which goes with the effluent. Now, once we have separated the sludge from the sedimentation tank, some portion of it is sent to waste sludge, and some portion is sent again back to the reactor, which is known as the recycle stream. Okay, so here since we are dividing both streams will have same concentration, right? So here you can say the concentrations are same, XR is same, but the flow rate or the amount may be different. Uh, Yeah, so can you tell me why are we providing the recycle to the reactor or the activated sludge process we are providing recycle? What are the advantages of this? Yes, Nidhi, are you there? Can you tell me? Okay, so let me tell you. Here we are providing whatever microbes that we are using here in the activated sludge process. So that activated sludge process uses microbes. Now microbes, yes, Nidhi.
No, I do not know that.
Okay, okay. Yeah, wait a minute. Okay, I will explain. Right, just wait a minute. Yeah, so whatever the microbes we are providing, so what happens to those microbes? These microbes can either grow by using organics as their food, right? And or these microbes can die after some time. So whatever the solids or sludge that is present inside this activated sludge process and whatever that has settled, there are some microorganisms which are living and some which are died. Now, so whatever they have died, they will not multiply and they will not add to any process. So, while we cannot differentiate the live or growing bacteria, organic organisms with the died one. So it is important to have some culture which is always added in the form of live microorganisms. So since some fraction of sludge will have live microorganisms, so that is recycled, while some might be dyed also, so we need to take out some amount every time. So that is why due to reduce the accumulation, the dyed bacteria microorganisms they needs to be taken out, and that is why need to have a waste sludge stream which is coming out and to ensure that there are living bacteria and organisms, we send a recycle stream, right? So here there are various aspects that you need to consider while designing or various design aspects. So one is F to M ratio, which is food to mass ratio. So this food to mass ratio, let me take a new slide. Yeah, so these terminologies are actually common for all the types of uh, biodegradable secondary treatment processes which involves microbial digestion. Okay, so food to mass ratio, it is known as F by M. It is the food present in reactor. Oh, sorry. Wait a minute, there is something wrong. Yeah, so what we were looking was F by M ratio, which is food in activated sludge process reactor or tank. Okay, let me write here only. Okay, so F by M equals to food in tank divided by mass of uh, microbes in tank. Right, so this is the food means here the organics divided by mass of microbes is the microorganism microbes quantity. So that can be found out by multiplying the volume into the concentration SE divided by, let's say, X is the microbial concentration uh, 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 into the amount. Okay, so into again the volume that has been taken up by microbes, right? Uh, yeah. So next term that is important is MLSS and MLBSS. So MLSS, it is the amount of solids present in tank. Now this solid can not only be because of the microorganisms present, but it could be because of some other uh, other suspended solids which are also present simultaneously along with the microbes that can result into some like so this is mixed liquor suspended solids. So mixed liquor is nothing but anything other than the suspended solids or these uh, other than suspended solid is mixed liquor. So ml here represents mixed liquor. Uh, so whatever the amount of suspended solids that like contributed by all is MLSS, while MLVSS is mixed liquor volatile suspended solids. So that is the amount of microbes present. Microbes present in the tank. Okay, so I hope you got the difference between MLSS and MLVSS, where MLSS contributes to all the solid present and MLVSS takes care of only microbes. Why MLVSS is important? Because our main aim is to have an optimum microbial concentration. So MLVSS has the prime importance in this process. Okay. Now next is sludge volume index. So it is a factor. Okay. Let me write here sludge volume index. So it's a factor that represents how well the sludge can settle. And how compact it can settle. So, for example, the SVI, sludge volume index, you can look into the formula and the derivation as well. So, here the criteria is within 30 minutes and in 1 liter cylinder, if you add a certain quantity which is prescribed in the protocol. So if you add that much quantity of wastewater containing sludge, so what time or what volume it occupies after 30 minutes of settling. right? So in that case, this is the definition. So smaller the sludge volume index, which means that uh, sludge has settled easier and in a more compact manner. Okay, so that is a preferred one, while uh, a higher sludge volume index, which means that poor settling properties of sludge, and it will float, and such uh, settling we do not usually prefer while uh, using this process. So again, the SVI index, which is ideally suitable for the given purpose, is given to you in the uh, notes. Yeah. So next factors that has been looked upon was the uh, mass balance across the activated sludge process, which will give you various parameters, including the retention time, cell retention time, and other parameters that can be derived. This can help into finding the efficiency of the process, right? So you need to write two types of mass balance here in the activated sludge process. One is for bacteria, microbes. Another is for the organics. Okay, so what is the mass balance generalized equation? Whatever is incoming minus whatever is outgoing plus the net rate of generation because of the growth and net rate minus the net rate of consumption because of the decay or some other reaction is equals to the accumulation. Now for continuous and steady state process, the accumulation becomes zero. So that is why on the right hand side, it is zero, right? So I'm not going into the details of the derivation now, but you can look that into later. Or you can, if you have any doubts, you can tell me. Otherwise, I will move forward. You can, like in the class, it is discussed in detail, so you can look into that. Yeah. So next, if we look into the types of activated sludge process, so it can be conventional, high rate, extended aeration. So now this depends on the rate of uh, hydraulic. Okay, so here one important parameter that you need to look into is hydraulic retention time. And another is mean cell residence time or cell retention time. So there are two types of retention time here mentioned. Can you tell me what is the difference between these two retention times and why two types of retention time are there? Or what difference is there? Yes. Okay, let me tell you. Okay, so here the hydraulic retention time refers to the retention time or retention time of wastewater in tank, and this is for the microbes in tank, right? So these two refers to, one is for wastewater, which is hydraulic, another is for microbes, which is microbes, or you can say the sludge in the tank, which is cell retention time. So depending on the retention time, if the rate is high, which means the retention time will be low, okay? In that case, efficiency will be low. While in case of extended aeration, higher retention time might be provided or would be provided. So it can give you bigger or higher uh, efficiencies. Next is that you need to consider is the amount of oxygen that will be required, right? So that can be supplied through uh, air, or you can simply have pure oxygen that can be supplied in the process, right? So what you can, by what ways you can supply the oxygen to a process such that uh, minimum, so okay, here you need to take care minimum energy requirement because you are using some diffusers, you are using some power to run those diffusers. You are using, if you are using pure oxygen, then you are using some cost to purify air. Okay, so such costs are also incurred with O2 supply in the process. So obviously you need to have optimum parameters for these also. So here for air supply, okay, so let's say this is the tank and this is the uh, recycle stream, this is the inlet fresh wastewater, and this is the actual inlet. So this is the length. Okay, so across the length, let's say um, uh, here it will be the maximum oxygen concentration required where it enters. In case you are not providing any stirring, okay, so whatever it goes, some sort of uh, uh, you know uh, mixing at small scale or not complete mixing will be uh, done. So in that case, what happens is the oxygen requirement will be maximum here at this entry point, while it reduces. While in the case of a conventional activated sludge process, we are providing a constant oxygen supply throughout. So which means that oxygen is not required that much. So in that case, if we are using this tapered, so what it does is, so uh, let's say the profile that oxygen requirement follows, similar oxygen profile of supply we also provide, right? Other than that, we can have some step input of oxygen, or we can have a complete mixing inside the tank such that uh, the oxygen requirement is uniform and you can have a uniform, like the concentration requirement will also, like this is required and this is supplied. Obviously you will supply some excess of oxygen, but so whatever, since it is completely mixed, uh, both profile will be linear. Okay, so this was all about the activated sludge process, which is the most conventional one. So this was discussed in detail. While another, the another type of uh, this secondary process that can be used could be trickling filter. Now, this could be aerobic or anaerobic. So depend. This is the image of trickling filter. Let's look here. So this is a packing, okay? Now this packing could be of rock, sand, or some high rate filters can have synthetic plastics as well. So this is a packing. Now wastewater is, so it is a closed chamber, unlike the activated sludge process which was having an open tank. Right, so this is the difference between activated sludge process, one of the difference between activated sludge process and um, this trickling filters. Now, since this is a closed chamber, it can perform both um, aerobic as well as anaerobic process. If depending on the supply of air, if it is made or not, right? So now let's look into the design. So here, these are the packings or filter media, and let's say this is the filter media. So on this, there will be microbes which are attached. Okay, so this filter media are nothing but support provided for microbes to attach on these filter media where they are attached, and wastewater, it flows through this. Okay, so it flows through this, and there will be some contact between. Uh, so here, water flows means it trickles down this media or this bed, right? So in that case, what happens is there will be a contact between uh, trick, sorry, microbes which are present onto filters and wastewater. So by this contact, the degradation occurs on the this filter media. Okay, so yeah. So here, so water flows down or trickles down this trickling filter, and this is the inlet where you this goes up, and it is through nozzle. It is sprayed, or there will be some flow, and air passes in the upward direction. Now, whatever the filtered water is there, it is coming out of the tank, and there could be some dead biomass also that can detach, or some live biomass also that can detach from the trickling filters. So, so once they have detached, they also need to be separated in the clarifier to have a settled sludge, right? And the treated water can be separated. And later, some recycle can be made to provide more uh, microbes. Yeah. So this was all about, and also the equations that are governing the size of the trickling filter and the efficiency of trickling filter. They are all mentioned in the trickling class. While it is very difficult because of the dynamic process, it is very difficult to derive, but whatever approximate values that you can have, is the formula is well mentioned in the class. Now, next type of uh, process could be uh, upflow anaerobic sludge blanket reactor. Okay, so here it is the upflow anaerobic sludge blanket reactor. Right, so what is that? Can you tell me? Okay, I just request you to wait for a minute. I will rejoin. Like, I'm just coming back in a minute. Please give me a moment. Okay, sorry for disturbance. Let's start back. Yeah, so here, what we are doing in this upflow anaerobic sludge blanket reactor, let's say this is, or let me just zoom this image to have a better visualization. Yeah. Okay, so let's say this is the inlet. Okay, and this is the sludge blanket. So this sludge blanket, uh, through this, through some nozzles and some uh, spray, influent, it goes up. Okay, so it passes through this sludge. Now this is an anaerobic process. Okay, so anaerobic means there is no oxygen supply. And since there is no oxygen supply, it will be a closed tank. Okay, so closed tank will be there because you do not want, because atmosphere also has air and which has 21% oxygen. Okay, so that you do not want, so oxygen interaction is not allowed. So you see here, this is a closed chamber. Now this sludge blanket is nothing but microbes plus some other suspended solids that could be there. Now this blanket, when water passes through this, microbes they degrade the organics present. Now here what degradation occurs? The organics. Okay, wait a moment. Sorry. Just a second. Okay, yeah. So due to the anaerobic conditions, this organics plus microbes, they will give you CH4 instead of CO2, right? So instead of CO2, uh, CH4 will be formed because of the anaerobic conditions. Uh, yeah, so now this CH4 has, the biogas plants. So these biogas plants, they are made to generate CH4. And you are generating CH4 here by some means, by the digestion or degradation of microbes. So in that case, what happens? You can collect this CH4 and later you can use this gas. Okay, so there is a gas collector here, and further that gas can also be purified because some water can be trapped with it that can be eliminated. So, you know, that gas will be collected and that will be taken for further use. So here, this is how it works. And the important formulas that you need to take care or look into uh, are sludge age, and mean cell age. Okay, so there is a difference between two. So age is nothing but just a second. Yeah, sludge age is nothing but total mass of the suspended solids in the basin per unit mass of the suspended solids, that is going inside the tank in the influent, while mean cell age is nothing but what total MLSS per unit solids that are wasted with effluent. Okay, so that is the difference between these two definitions. Okay. Now, another type of system that we can use in secondary process are lagoons, where lagoons are nothing but uh, microbes are provided, but there is no aeration provided. Okay, so in that case, can you tell me how, like this is the image and this is the inlet. So can you tell me how the process occurs and what type of process occurs in case of lagoons? Yes, anyone? Nidhi, Manas, can you tell me or explain the lagoon process? Okay, let me tell you. So let's say this is the, there's the pen. Yeah, so this is the inlet and this is the surface which is in contact with atmosphere. So here the lagoon systems, they are utilizing the oxygen which is present in the atmosphere. So oxygen interaction will be there. So oxygen will be taken up by wastewater, and as the organics they are degraded, so there will be a concentration gradient across this. So oxygen will be taken up. So here it will be maximum oxygen concentration. It will be taken up, and there will be a point where oxygen concentration is not there. Okay, so after that point, so let's say this is that point. So after this point, the process gets converted into anaerobic process because there is no oxygen supply. Further, this oxygen uptake process is also dynamic process because it continuously goes on. So why it goes on? Because the organics are continuously getting degraded by microbes by consuming oxygen. Okay, so there is a decrease, constant decrease in O2 concentration also in the aerobic zone also. So, but this zone is actually in contact with the atmosphere from where it can take up oxygen, and that is how you can say a particular zone there will be aerobic zone, while in um, another zone there will be some portion where anaerobic conditions will be prevailing. Right, so this is about lagoons. Now, next is some advanced processes. So these were all nothing but conventional process, out of which activated sludge process is the most common one. But one of the major drawback of activated sludge process is the high space requirement because it requires multiple uh, tanks for activated sludge process, like the aeration tank and which is followed by secondary sedimentation tank. So instead of that large space requirement, so if you can have all the process in one tank, so that is performed in sequential batch reactor. Okay, so sequential batch reactor means sequence of process taking place in batch mode in a single tank. Okay, so here this is important. So single tank is used in case of sequential batch reactor. So can you tell me how these batch process and continuous process, like what is the basic difference between the two? and how in industries they are followed or they are performed. Can you tell me? Yeah, so let me tell you. So batch processes are nothing but whatever you have. Let's say you have um, wastewater volume V. Okay, so you dump all the wastewater which is having volume V, and for that microbes required, let's say it is having concentration X, so that much microbe seed you provide. So X is the microbial concentration. So that much is provided already at time t equal to 0. You add or you dump everything. Now, depending on the conditions required, you might provide or might not provide stirring. But as the time progresses, now once you have dumped all the inlet or raw materials to that tank, so this is a reactor. So once you have dumped all the raw materials or reactants inside that reactor, so after that there is no supply. But as the time progresses, because of the presence of all the raw materials here in the case of wastewater treatment, the organics and microbes are present. So as the time progresses, organics degrade, and this is a kind of um, time driven process, so variable time process. So as time progresses, the process goes further or process moves ahead further, and the efficiency increases, and at the end the process ends after some time, so variable in time. While in case of continuous process, there will be a continuous inlet, continuous outlet, which is there, continuous stirring and reaction taking place. So the concentration, whichever is inside, that will be coming out. So that is a continuous process, um, no, taking place. So here, what you are using a batch reactor, and only a single tank. So there only you will follow uh, multiple steps in the same tank. So where you will first add all the wastewater. Okay, so first and foremost thing required is you dump all the things that are required. So you dump or you add the wastewater. So that is filling of the tank. Now that after the filling, uh, there could be different ways of it, where the filling could be static. Okay, so there is no mixing. There is no external mixing through propellers, while another could be there is some mixing provided to keep all the microbes uniform throughout the tank. So that is mixed, but you are not providing any aeration. While there is last aerated also, where you are providing aeration as well. Okay, so depending on the conditions and the type of wastewater, the amounts of BOD, COD, this all parameters they actually depend on the BOD and COD, because the activated sludge process is not suitable for the processes which are having very high BODs. Okay, so it fails to work at very high BODs. So in that case, you need to have, so depending on the concentration of BOD, whether aerobic conditions are required or not. So um, if BOD level exceeds a point, then you need to have anaerobic process. So what sort of aerobic conditions you also need to have? Depending on that, you provide all these conditions. Next is you allow it to react. Okay, so for some time reaction will be taking place. Now there could be some anoxic or aerobic condition might be prevailing. And then once the reaction has occurred or the degradation of organics has occurred, taken place, you allow them to settle. Okay, so sludge needs to be settled because in activated sludge process, so this sequential batch reactor is nothing but activated sludge process. So SBR is nothing but ASP in batch mode in just one tank. Okay. So here in the activated sludge process, you had one aeration tank followed by one sedimentation tank. So here, since we are doing all the task in one tank, after the reaction, we are allowing it to settle. Now that after settling, there will be some layer of water which has come up. So that is known as decantation, and sludge has settled down. So you can decant or you can take out the treated water. So that is decantation. And there is some idle time which is provided to because of such long processes. Some idle time provided in between two subsequent or two back-to-back process. Okay. So the total time of this batch reactor taken will be summation of all the time. So what time is required for filling the tank, what time of reaction you are providing, what settling time you are providing, what decantation time, and what idle time you are providing. So that is nothing but cycle time, which is summation of all the times which is required. Then comes hydraulic retention time and the solid retention time. Now here there is a tricky point. So for retention time, you take the basis of, okay, so you take volume per unit flow rate. But here there is no flow rate. Okay, you are just dumping all the wastewater at a time. Okay, so what you do, you consider this flow rate by whatever the amount of water you are treating in one day. Okay, so that will be nothing but wastewater treated per day. So by that you can find out. So volume treated per day. So that will be nothing but meter cube per day. So that is nothing but a way of representing flow rate. Okay. Anyways, flow rate is nothing but Q, which is inlet wastewater treated per day. Okay. Or inlet wastewater in meter cubes per day. So here also we are finding a way to find out meter cube per day. So this Q is um, wastewater treated daily on daily basis or per day basis, right? Yeah, so this was about sequential batch reactors. Now there could be some multiple processes that might be happening in the sequential batch reactor. So apart from the aerobic digestion or there could be nitrification, denitrification process also going on along with the degradation process, where during anoxic condition denitrification occurs, while during aerobic condition nitrification occurs. So that you need to remember because whatever the ammonical nitrogen is present, so in the presence of oxygen, this gets converted to nitrites and nitrates. That is nothing but nitrification. While during this denitrification, there are nitrates, nitrites which are already there. That could be during the absence of oxygen that can be converted to N2. So that is nothing but denitrification, right? So that also depending on the amounts of nitrogen levels present in the tank and the oxygen conditions prevailing, that process also can occur because excess of nitrogen is also harmful for the water bodies. Okay, so so this process simultaneously removes nitrogen as well. Okay, so why or it removes nitrogen through denitrification. So can you tell me the process name? Why nitrogen is harmful uh, if it is in excess? Can you tell me? Yes, Manas, are you there?
Yes, I'm here.
Yeah, so can you tell me why we need to have nitrification, denitrification, and what are the drawbacks like in case if we have more concentrations of nitrogen in wastewater and if such water is dumped in any of the water bodies, then what could be the drawback that we might face? So, what is the disadvantage of it or limitation of having higher nitrogen concentrations? Do you know?
No, ma'am, I have no idea about.
Yeah, okay, so let me tell. Okay, so you might be knowing that for the growth of plants, the fertilizers that are provided, it consists of nitrogen, phosphorus, and potassium, right? So that is nothing but a nutrient which is required for the growth of any plant, okay, or any crop. Now, if such high nitrogen rich wastewater it ends up into water bodies. So, there are seaweeds, there are algae that grow in water bodies. It could be in lake, it could be in river as well. So, if you dump such high concentration of nitrogen containing wastewater in water bodies, the algae will grow, and what happens is they consume oxygen for their growth, right? So oxygen will be taken up for the growth of those plants, and also there will be some degradation that might simultaneously grow on. And further, there are chances that let me show you the picture of it. By that you can visualize the process. Okay, so you see uh, here the after certain point the algae that has grown or some of the plants that has grown, it covers all the surface of water. So for the breathing of this uh, aquatic animals, the oxygen which comes, it comes from the atmosphere. Now, since the layer of plant has covered the entire water body, so oxygen will not be able to the dissolved oxygen concentration reduces because the contact between air um, reduces. Also, the sunlight which is required uh, by the aquatic bodies, there will be certain sunlight also required. That will also be lesser available, right? So this type of entire coverage might be present in the water bodies that might result into some adverse impacts on the living organisms because of the limited dissolved oxygen that can result due to higher nitrogen concentration. So that process is nothing but known as eutrophication. So obviously that is why we need less or proper nitrogen concentration or removal of nitrogen also in case if it is in higher concentration. Now next is so far what we have seen is conventional and sequential batch reactor. Now here the microbes that are there, they are very small. Okay, so some special sedimentation processes required and which requires like smaller the size, more time it will take for settling, right? So in other case, if we somehow increase the sizes of those microbes, then in that case the settling will be faster. So we are by increasing these time or by reduce sorry not increasing the time by making the process faster what we are doing we are reducing the time required for the process and we here we can also reduce the cost by having a smaller tanks. Okay, so that is taken care of in the granular activated sludge process, where microbes they are used in a bigger in the form of granules and instead of small flocks of microbes. So let's say this is the granule of microbe. So what type of process occurs? So on the surface there are oxygen contact. There is oxygen contact available. So here there will be some zone where aerobic digestion occurs. Now further there will be an anoxic zone where the oxygen will be provided by the denitrification. Okay, so here the nitrification occurs first place due to the availability of oxygen. Okay, so this is the nitrification zone along with the digestion and degradation of organics. So there will be two processes: nitrification and organics degradation. Right, so two processes simultaneously. Now there will be a layer after which there will be limited oxygen supply or no oxygen supply. So in that case, anoxic layer is present where denitrification occurs. So whatever the nitrification has occurred, so nitrites and nitrates that are present, they get converted to N2 through denitrification, and the degradation occurs of the organics. While in the inside top, most layer, again, this also denitrification is also absent. So such layer is anaerobic core, where anaerobic process or degradation through anaerobic process occurs. Now, what is the advantage of using such uh, granules? So it helps in faster settling. So you will have faster settling of the microbes. And how do we have such granules? So for that you need to have days of aging of those microbes where extracellular uh, uh, mass or some extracellular polymers or product they are secreted, which results into uh, attachment of microbes firstly through some van der Waals force or some interaction force. They come closer, and the ESP that is secreted by these microbes, they also further help in growing. They keep the microbes to be come in contact and also grow. Okay, so it will grow bigger and bigger because of that ESP that binds it, right? So that is nothing but granular sludge process. Next is the membrane bioreactor. Since this is a sort of digestion or degradation process, so let's say this is the tank of that degradation process. Now instead of sedimentation tank, okay, so this is sedimentation tank. So instead of this sedimentation tank, you are providing a membrane where you will have filtration of microbes from water. So water will be filtered through uh, the membrane. So membrane is nothing but example of, for example, uh, the RO process. So those are also nothing but membranes which are used in the household. So similar membrane you can have one tank or you can have two tanks for using membrane um, in which is clubbed with the uh, digestion process. Now let's say let's see next type of treatment processes where we can have some uptake, or these are nothing but like not so conventional, but these are very some recent processes which are used for some contaminants. So this is a phytoremediation process where we use some right? Now, these plants they take up the pollutants, and there are different mechanisms. First is phytostabilization. Okay, so what plant does? Okay, so plant can take up the pollutant which are present in the soil, or it can take up the pollutant which is present in groundwater. So, what does? So, let's say this is the ground, this is the soil layer, and this is the tree which has the ability to take uh, or absorb this pollutant which is present, and these are the roots of that plant. So now this roots they have the ability to uh, concentrate or to limit its mobility by some sort of maybe conversion of, um, let's say some pollutant CR6 is present. This is a pollutant which is heavy metal, right? So now if this plant has ability to convert it into CR3. Now CR3 is less toxic as compared to CR6 because this is less mobile. Now less mobility means it is accessible lesser to other organisms, and hence it is lesser toxic. So such sort of phytostabilization where mobility, so this is just one example, there could be some other examples as well, where the mobility of pollutants is reduced by some conversion or by some attraction. Okay, so that is phytostabilization. Then phytoremediation. So in that case, the roots of the plant or the tree that takes up the pollutant, so and that will be stored here in the plant itself. Now another could be rhizosphere bioremediation, where the roots it secretes the certain material or certain enzymes, you can say, that helps in the remediation or conversion or degradation of the pollutant which are present. So that sort of, it enhances, it do not take up, but it enhances the degradation or remediation by secreting enzymes or some other materials. Then filtration is nothing but if the roots they take up um, the pollutants from the soil, they are transferred further into the stem um, and other part of the plant. And last is nothing but the phytotransport transformation, where the pollutants they are taken up and also further they are degraded or they are transformed in some other form, and most probably they are transformed into the forms that are lesser toxic. So it is a kind of very environment friendly process where you are using plants for remediation. So it will there will be a plant coverage. By this way there will be an increased plant coverage, tree coverage on the earth, and also there will be a simultaneous reduction in the pollution. So that is very one of the most common advantage, while it takes a longer time for the process to occur, so it is a kind of slow process. You need to have patience for this process. So bioconcentration factor for this process, if you look into, it is the how much ion accumulation ability the plant has. Okay, so as compared to the soil concentration, what is the concentration in the plant? While in the case of translocation factor, it is for a per unit amount of pollutant which is taken up by the roots of the plant. What is the amount which is transferred in the entire plant? Okay, so let's say it is transferred into the stem or in the leaves of the plant. So what is the uh, internal transportation of that um, you know pollutant? So by that way, if you can say if that translocation factor, if let's say it is very small, so there will be a capacity of the plant also or of the tree also to sustain that particular pollutant. Okay, so there are number of plants which are having, you can see the look into the list of it, and there are specific trees and plants which are specific towards a particular pollutant. Okay, so if the pollutant concentrates at a particular portion of the plant, then there are chances that once the limit has crossed, there could be some adverse impacts also to the plant, so which we do not want. So in that case, if the translocation factor is more, that plant can take up more. Okay. So now next type of some similar environment friendly process is phycoremediation, where you are using macro or micro algae for the reduction or biotransformation of pollutants. Now, what happens here is, in the presence of sunlight, the algae, it takes up carbon dioxide and it emits oxygen. Okay, so it is a sort of plant which requires carbon dioxide and it generates oxygen. So now this oxygen is taken up by microbes or bacteria for the degradation of the organics which are present. So there will be a zone which is, let's say there will be some zone till this point which is aerobic because of the oxygen which is provided by the algae. Now this bacteria also degrades the this uh, organics. Now that generates CO2. Now there is a cyclic process where algae grows, it takes up CO2 by that is generated by the degradation of organics, and it further grows by releasing oxygen that is further taken up by bacteria. So that is a cyclic process. Obviously there will be a zone because that oxygen, if you are not providing any external mixing, so there will be a zone where oxygen is not reaching. So there will be an anaerobic degradation zone as well, where CH4 also is generated. So this is also you are using some, you are not providing any external means for degradation. So it is also an environment friendly or more sustainable method as compared to the conventional methods where you need to have constant oxygen supply, constant, some supply of electricity will also be required. So, this sort of processes are also sustainable in nature. I hope it is clear so far. Then we can move to the assignment part. Yes, Manas?
Yes, ma'am. Yes, ma'am.
Okay. So let's move to the assignment. So, which of the following statement is true about the high rate activated sludge process? So here you need to look into the key terms in the question, which is high rate. Okay, so high rate means shorter, means the rate of process is high, which means you are providing shorter time inside. High rate means, sorry, high rate means faster flow rate, and faster flow rate means with same amount of uh, or same tank size, so the retention time, which is volume per unit flow rate, so here the flow rate increases, which means for same volume of tank you have short aeration period. Okay, so high rate of activated sludge process means shorter aeration period. Okay, and shorter aeration period, along with that it results into higher sludge production as well. Okay, so this is the correct option here. uh, yeah. So next question is for a good sludge, the value of sludge volume index should be in the range of dash. So this is a thumb rule which is given to you in the lecture notes, which is 40 to 100, I guess, right? So sludge volume index is the index which gives you the settling ability of sludge. Okay, so lesser the amount of sludge volume. Okay, so sorry, this is the amount of volume sort of this index. It represents the how much amount of volume after settling the sludge occupies. Okay, so more the volume sludge occupies, it has it is, you can say it is dispersed in the volume of water. Okay, so it is dispersed in water. So lesser the volume of water it has taken up, more is the settling ability, you can say. So smaller values are recommended. Next question is, which of the following material is more preferable as a packaging material or packaging media in a super rare filtering filter? Uh, it is a very simple question. Can you tell me? Okay, so the answer to this question is synthetic plastic. So because sand, stones, and so these are the conventional materials which are used in triplet filters. Now, this synthetic plastic they have such dimensions so that the rate of process can be enhanced. Okay, so these are specific packaging material. So these are next we have got sequential batch reactor from sequence of steps. The reactor goes through as the reactor goes through as it collects wastewater, treats it, and then discharges all the steps are in batch tanks. So this gives suggest sequence of process working on the steps are followed at the same time. Okay, so it goes one by one in the same tank. So a single tank is only used in sequential batch reactor. So name itself suggests here you need to just completely to the sequential batch reactor. So all the steps performed in a single tank. Next question is preparation of sequential batch reactor. The middle step occurs between decant, step, and fish phase of the next cycle. So let's write down the sequential batch reactor steps. So first is the fill step, which is filling of the wastewater. Now once the tank is filled up by wastewater, allow it to react. So reaction takes place between the microbes and the organics, where microbes grow, organics is consumed and degraded, and the reaction completes, after which you need to separate the starch through settling process. So settling or settling process occurs, right? And next process, once the settling has occurred, so you need to decant it where you are separating the treated sludge that has settled. And next comes the idle step, which is the last step for the cycle. So and after the idle step, we have to that. So the correct answer to this question is fill. So after the idle step, the next process that is followed is fill. So next question is solid retention time determines the time for which the biomass is retained in the reactor, right? Now the SRT for the SBR, which is sequential which can be converted by 24 by TC, because this TC is in hours and this NC is need to be calculated in days. Okay, so you substitute this. So if you substitute this by 24 by TC, then you will have 24 by TC, which will be VTXTC divided by VW XW into 24. So here the option is A, right? So it's very simple. You can actually derive the formula very easily. Now next question is, which of the following is the type of open photobioreactors? So the correct answer to this question is raceway pound. Okay, and because other options you see, uh, uh, they are all closed ones. Okay, so let's move to the next question. So can you identify the type of photobioreactor? This type of this is very simple. Filters that are there, they are optical tubes. So check the this match, which is vertical column. These are columns. Next is a numerical that you need to solve. So let's look into that. So you have a complete mixture activated sludge process. So this is the activated sludge process tank, which is having complete mixing. And to that, 2 mld. Now this mld means 2 million liters of wastewater per day. It is flowing into the tank, which is having BOD. So S note is having 250 milligrams per liter, and the biomass concentration inside the aeration tank X is 2500 milligrams per liter. The concentration of net biomass leaving the system per day XE is 70 milligrams per liter, and the aeration tank has volume 250 meter cubes. Now you need to find out the hydraulic retention time of wastewater in the aeration tank. So, yeah, Manas, can you tell me what is hydraulic retention time? Okay, let me tell you. Um, there are two types of retention times that, like I have discussed previously in the previous slide itself. So there are two types of retention time for any aeration tank. One is hydraulic retention time. That is for the that represents the amount of retention time or the amount of water uh, that spends time or the amount of time water spends inside the tank, while another retention time is for sludge. Okay, how much time sludge spends inside the tank. So here we need to find hydraulic retention time, which is nothing but the HRT, which is volume of wastewater, volume of tank per unit flow rate. So this is a very simple formula for activated sludge process, and you have direct values which are given to you. So volume of tank is 250 meter cube. Now the flow rate that is given to you, Q, is 2 mld, which is 2 into 10 to the power. So this mld represents here million liters per day, right? Okay, so million means 10 to the power 6 liters per day, right? Now this is in liters while this is in meter cube, so you need to have unit consistency. Let's convert this liters into meter cube. You can do otherwise also, everything will be one and the same. So 2 into 10 to the power 6, so 1 liter is equals to 10 to the power minus 3 meter cube. So multiply by that factor, so this will be meter cube per day. So what remains is 2000 meter cube per day is the flow rate of wastewater that goes into the tank. Sorry for this. Yeah, so if you substitute the values, you have to substitute here 2000 meter cube per day. So this will give you, you need to solve 25 by 200, which will give you, Manas, are you following? The answer will be
I'm
0.125.
following, ma'am.
So meter cube will get cancelled and this day will go upwards, which will be 0.125 days. Right, so options are also in days. So correct answer here is 0.125 days. Yeah, you followed it, right? I could not get you what you told. Sorry.
Yes, ma'am. I'm following, ma'am.
Yeah. Sorry, I could not hear you properly. Thank you. Yeah.
Thank you,
So,
ma'am. Thank
yeah. Let's move to next question. This again is a numerical. And you might expect similar sort of numericals in your assignment and maybe in the exams. Yeah. So a completely mixed activated sludge process, again similar activated sludge process, where you have a wastewater which is flowing at 550 meter cube per hour flow rate, which is having BOD S not equal to 200 mg per liter, right? The concentration of soluble BOD which is going out, so BOD going out which is SE 20 milligram per liter. Now the design criteria are as follows. The yield is zero point 0.5, K is zero point 0.5 per day, KT which is the decay constant
0.05 per day and Ks is 80 mg per liter. MLVSS or X concentration is given to you as 2000 milligram per liter. So this X is inside the tank. You need to find out the mean cell residence time theta C. Okay, so let's see what is the formula for that. Okay, so this is the formula, but this theta C is inverse of theta C, but I guess here it has to be SE. So it is YK SE by KS plus SE minus KD. This should be the formula for 1 by theta C. Okay, so let's find out 1 by theta C by just putting up all the values, because you know Y, you know K, you know SE, you know Ks, and you know Kd, right? You know all the values. So by that you will get the inverse of mean cell residence time. And once you do the reciprocal, you will get theta C. Okay, so let's first find out 1 by theta C. Put up all the values. Zero point 0.5 is Y. K value is given to you as 5. SE is 20. KS is given to you as 80 plus SE is 20 minus KD is 0.05. Solve this, you will get 1 by theta C value. Yeah, so 0.5 into 5 into 20 divided by 100, right? So it is 0.5, this first term, minus 0.05, which will be 0.45. Now this is the value of 1 by theta c. Now to find out theta c, take the inverse of 0.15, which is 2.22 days. Sorry, 2.22. Now this value you are getting is in day inverse. Okay, you see, because the KD value you see here, it is in day inverse. So all the resultant value that you will have will be in day inverse. So 0.5 day inverse. Okay, so if you take the reciprocal of it, it will be 2.22 days. Is it clear? So should I discuss the tutorial questions? Do you want me to discuss? Then in brief, whatever remaining time we can, whatever we can discuss, I can discuss. Otherwise, we can end this meeting also, because tutorial has been completely discussed in the class. So whatever you suggest, we can do. Yes, do you want me to discuss? Ma'am, just one question to me. Hello. Yeah, ma'am. Actually, during exam, what kind of questions may come, and for MCQ or the broad or descriptive type? Ma'am, what type of questions, and only key MCQ model questions now maybe come for the exam?
Now, actually, questions like as we saw in the assignment, there might be some numericals which you need, so you not directly have to take mark the answer.
Okay,
So
okay,
these
okay,
are
okay.
sort of questions
question.
Sure, sure. Thank, thank you.
Okay. So do you want the tutorial to be discussed in brief, or should I end this meeting?
I think this is enough for me for today.
Okay,
Uh,
okay. Thank you. So then we
okay.
can end this meeting for today. We will meet in the next class.
Okay. Okay.
Thank you.
Thank you.
Thank you for joining.
Thank you.